Welcome to the Scientific Sense podcast, where we explore emerging ideas from science, policy, economics, and technology. My name is Gil Eapen. We talk with world's leading academics and experts about their recent research or general areas of topical interest. Scientific Sense is an unstructured conversation with no agenda or preparation. We cover a wide variety of domains where new discoveries are made and new technologies are developed on a daily basis. We are most interested in how new ideas affect society and help educate the world how to pursue a rewarding and enjoyable life rooted in science, logic, and information. We seek knowledge without boundaries or constraints and provide unedited content of conversations with researchers and leaders who love what they do. A companion blog to this podcast can be found at scientificsense.com, and this podcast is available on over a dozen platforms and directly at scientificsense.net. If you have suggestions for topics, guests, and other ideas, please send them to info@scientificsense.com, and I can be reached at gil@eapen.info. My guest today is Professor Sue Gore, who is Professor of Bioengineering and Therapeutic Sciences at the University of California, San Francisco. Her research is on understanding how the brain works, connecting molecules to systems. Welcome, Sue.
Thanks for having me, Gil.
Yeah, thanks for doing this. So I want to start with one of your older papers from 2011. It's entitled Toward Molecular Genetic Dissection of Neural Circuits for Emotional and Motivational Behaviors. You say in this paper, you ask, how does the brain process the emotional meaning of sensory stimuli and in turn drive behavior? Studies in the mammalian systems have identified various brain regions and neurotransmitter systems that are critical for emotional and motivational behaviors, and have implicated their involvement in neuropsychiatric disorders, including anxiety, depression, schizophrenia, and addiction. Despite these significant advancements, you say the precise neural circuitry underlying emotional and motivational behaviors remains to be understood at molecular and cellular levels. So this has always been the problem, right, for neuroscience. The hardware seems just a bit too complicated. And so what is your sort of focus in this paper?
Yeah, so that is a review paper we wrote, almost 10 years ago. So I'm very interested in the brain ever since I got my PhD. So I always wonder, how does the brain actually process, especially emotional information? So like, how do we, certain sensory stimuli will make us, right? For example, if we see an old friend, or if we're really hungry and then see, a delicious hamburger, right? So we'll feel really happy, right? So how does this certain type of sensory stimuli can elicit, emotional information, emotional feelings inside ourselves, right? So, and whereas some other sensory information is pretty much neutral, right? Right. It doesn't really trigger a lot of the deep emotions. So I'm really curious in understanding what differentiate these sensory stimuli, what makes them, tick our brain in a different way.
Yeah, and that is truly complex, right? So we make some advancements in artificial intelligence, and when we think about machines, it tends to be rather prescriptive. So we have inputs and outputs, but when we think about emotions, that is a very, very complex phenomenon, right? As you say in the introduction here, lot of brain areas get involved in this process. So really understanding how it works is really challenging, I would think.
Yes, yeah, yes, you're absolutely right. So, speaking of AI, so I remember once I watched a TV show like on an airplane. So they were talking about these robots, right? So how the robots can perform tasks given, right? However, they don't currently, at least, can sense the emotional, justice or things like that, right? So how do you program a robot to be, compassionate, right, and then be able to have this type of emotions? Right now is I don't think it's possible. Speaking of the complexity, yeah, absolutely, you're right. Our brain is thinking about human brain, for example, is extremely complex. So that's why we really, wanted to study a simpler brain, if you will. So that is the zebrafish brain that our lab is focused on.
Hmm.
Yeah, so the human brain has 100 billion neurons with like trillions of connections, whereas the little tiny zebrafish brain has many orders of magnitude fewer neurons. Yet these creatures, they can display a lot of the same type of needs that we have. For example, they need to avoid predators, or they need to seek food as a reward, right? So these type of this very basic emotional elements are preserved in the simpler brains like zebrafish.
Yeah, that's really interesting. So just to get the magnitude of this, so the human brain, 10 to the power 11 neurons, 10 to the power 15
Mhm.
connections approximately, right?
Yes.
Whereas a zebrafish, you said 100,000, so 10 to the 5. So
Mhm.
yeah, it's about 6 orders of magnitude less,
Mhm.
the sort of the quantity of neurons in the brain, 6 orders of magnitude less. But you're saying even in that type of a scale, we are seeing emotions.
Yes, yes. So we have been study these lava zebrafish brain, right? So so the lava zebrafish brain have about 100,000 neurons, and these they also have a transparent brain, which is really great for imaging purposes. Yeah. So yeah, so we actually started with a very simple observation. So when we put this tiny little lava zebrafish, right, in a behavior chamber, and then we happen to put half of the chamber, right, half one side of the chamber to have a more illuminated, right, the other half of the chamber is less illuminated, is dark, right? So one side is light, the other side is dark. And then when we put these little zebrafish in there,
Yeah.
we can see that they show a distinct preference for the light side. Yeah, so they would avoid the dark side. You can even see them, seem to be very, a very choice making like process. So they would, obviously all creatures are very, like to explore, right? If they have the opportunity, they like to, go around the entire place.
Right.
But these lava zebrafish, in general, right, they would go to the border between the light side and the dark side, and then you will see them actually turn away from, to avoid the dark side.
Is
So
it
go
uh,
ahead. Mm-hmm.
so is it instinctual or is it emotional? For instance, if the the more lighted side gives the zebrafish a higher chance of seeing the predator earlier, take evasive actions. Is it sort of instinctual, or is you think they are actually making conscious decisions?
Yeah, Gil, that's a really, really great question. So, I'm not sure whether, whether the fish have consciousness, right? Or I don't know whether they do or they don't, right? So this behavior definitely is considered, we call it an innate behavior. So in a way that they seem like they know this is what they are supposed to do. It's not like learned from somewhere. Or from someone, yeah. So whether it's emotional or not, so I'm not sure whether emotional behavior has to be conscious, right? So we often think that emotions are sort of deep within, like subcortical controlled, right? So more have this sort of below conscious level of operation. But I don't know exactly how it works in humans. But we do believe that this behavior is not a simple reflex in larva zebrafish. So it involves processes and the neurotransmitter types that are really that can modulate a circuit. So rather than just the sensory motor, direct sensory motor.
Do we see the behavior in all stages of the zebrafish's development right from the beginning, or in other words, is there any information that tells us it's a learned behavior in some way?
Yeah, that's a really interesting question. So we believe that the larva zebrafish, so we do this behavior when they are just about 5 or 6 days old, right? So they're just like very young, and then we usually house them individually, right? So they don't really have an opportunity to learn from others in terms of performing this behavior. So we think this behavior is unlearned. So they're born to have this behavior. So that's the sort of the innate behavior. However, this behavior is completely modifiable by their experience. So if we sort of introduce them right into this light dark chamber repeatedly, right, and we can clearly see that they grow less and less sort of quote afraid of the dark side.
Yeah. So is there any connection to the visual systems? The preference for the more lighted side, is it because sensitivity to the eye, to the visual systems in any way, or that is not really there?
That's a really great question. So definitely this behavior requires an intact vision. So if we have, for example, larva zebrafish that are visually impaired, and then they will not perform this behavior very well. Yeah. So vision is required for them to actually make a clear choice here. So whether this behavior is entirely driven by like visual sensitivity only, I don't think so, because what's really interesting is we have been treating these larva zebrafish with drugs. For example, commonly used anxiolytic drugs in humans, like benzodiazepine, for example, which is a GABA agonist, or the buspirone, which is believed to act through serotonin system. So both type of these drugs can significantly decrease their dark avoidance behavior. So suggesting this behavior is maybe fear or anxiety related in this very simple organism.
Yeah, that's amazing. So you have a term here, conditioned place preference, CPP. So you say CPP behaviors have been widely observed in rodent models in the context of studying drug abuse and addiction. So what exactly is CPP?
Yeah, that's a really interesting question. So CPP stands for conditioned place preference behavior. So in this behavior, CPP is different from what I just talked with you about, which is the innate innate preference behavior, right? So CPP is actually a learned behavior. So it's conditioned place preference. So the way how it works is that so you would first introduce animal into a behavior chamber, right? That potentially have like 2 or 3 distinct compartments. And then initially the animal will show no preference for those compartments, right? So they have like a neutral preference, equal preference. And then what you do is you compare the animal with one particular compartment, right? So let's call it like the left compartment, right?
Yeah.
With drugs, for example, or anything that's really rewarding, for example, right? So then over time, so the animal will develop a preference for the left chamber, right? So, and then, sometime later, maybe 2 days or 3 days later, right? Then you reintroduce the animal into this chamber, right? At this time, without any rewarding stimuli on board, right? So just the animal and the chamber, right? And then you measure its preference. So now, if the animal has learned, right, the left chamber, for example, predicts, right, where it gets the reward before, right? So that now it basically predicts where the reward will be. Then you will find that the animal will spend a lot more time on the left chamber. So now that we can say, aha, this animal has learned, right, has developed this conditioned place preference.
Yeah. Yeah, so that is the learned behavior. So going back to the zebrafish experiment, you say that you do this very early, so it is not a learned behavior. 100K neurons, from a mechanistic perspective, we have deep learning networks that are much, much bigger than 100K neurons.
Mm-hmm.
So I'm just thinking that, so suppose, obviously it's not this simplistic, but, so suppose we take a deep neural network and we train that to dark images and light images, and it will be able to differentiate between the two mechanistically pretty quickly. Now the zebrafish, you say that the larval zebrafish, so it's very early in its progression. So it's almost like the brain is coming with some preloaded software, like an operating system. Yeah, with certain. I'm just making a statement, so you can correct me.
Yeah. That's a really interesting comparison. Yeah. So whether the larva zebrafish have preloaded software, software, right, that guides their behavior already before sort of any learning is taking place. I think that is the case, because these little creatures, right? If we think about their living conditions in the wild, right?
Yeah.
So they're faced with a lot of challenges, right? So their big fish potentially will eat them, right? And then they also need to survive and need to find food, right? So I think they need to be preloaded with a lot of softwares. Otherwise, the species probably will have gone extinct.
Yeah, and so it's clearly from a selection perspective, evolutionarily from a selection perspective, whoever collected good software were able to pass that on, and over time, and so when you're born, you already have some level of software in the brain. Now, is there a difference between what we think about as instincts and this type of behavior, or you don't see a difference?
Yeah. So I usually like to call this type of behavior innate behavior, right? So or sometimes we also call it like instinctive behavior.
Yeah.
But that is different from like a reflex behaviors, right? So reflex, for example, like the knee jerk reflex, like if you hit your kneecap, like your knee will automatically jerk, right? So that is a very simple reflex. So it basically go directly from sensory, right, to motor.
Yeah.
So there's not much information processing in between, right? So you
It doesn't even get to the brain, I think, right?
Right, exactly. We have peripheral reflex, right? Peripheral reflex behaviors. It doesn't even need the brain, right? So it's probably just very local, right?
Yeah.
Whereas instinctive behaviors is different from reflex. So instinctive behavior meaning that somehow you don't have to be taught, right, to do certain things,
Right.
and yet some of these things could be a complex procedure. Like it doesn't have to be like a reflex behavior. Yeah.
Yeah. Was there any sort of experiment so that so we know this is their sort of expected mode of operation? Is there an experiment where when if you were to give some sort of negative reinforcement for the lighter side, more lighted side, uh,
Mm-hmm.
can you reprogram the zebrafish to start to like the darker side over time?
Yeah, that's a great question. We have not done that yet, but we have basically observed the other way around. Is basically if we leave the animal, right, larva zebrafish, in the chamber for longer time, right? So and then they would, any creatures are naturally very likes to explore, right? So we think that they wanted to explore the entire chamber, entire arena. However, they're afraid to do so on the dark side, right? Initially, but still sometimes they will take a quick excursion into the dark side and quickly come back to the light side, right? So initially we find that they find dark side to be very very aversive, right? But
Yeah.
as time goes, right, if we let them be in the chamber for longer time, and then they will gradually learn that the dark side is not as scary as they initially thought, right? So then they can actually gradually spend more time on the dark side. So this already it's kind of like experience dependent learning is already happening. Yeah.
Yeah, so I mean, humans are sort of the same, right? So,
Mm-hmm.
sort of, I think we have an innate or instinctual aversion to darkness, and over time, we get used to it. As we,
Mm-hmm.
you say in this review article, so that in the conclusions, you see some implications for human neuropsychiatric disorders. You say for zebrafish, the world is a very different place from the one that we experience. Despite these differences, the basic needs that they have for feeding, sheltering, and reproduction are strikingly similar. So how does this what is the connection you see to the human neuropsychiatric disorders in this context?
Yes. Yeah. So we think that so this dark avoidance behavior, right, potentially are fear and anxiety related, right, in zebrafish. So the it's sort of a balance between how much they want to explore, right,
Yeah.
and versus how much they want to seek safety, right. So I think with anxiety disorders, usually it's characterized as having excessive fear, right?
Yeah.
Or uncontrollable fear of things that are potentially not as fearful, right? So we believe that the connection with human psychiatric disorders are potentially in the arena of anxiety like disorders or depression. Yeah. So and as yeah, go ahead.
And so, if the zebrafish is going to the dark side, mechanistically there are some neurons firing, and the net effect of those neurons firing is sort of anxiety and avoidance of that modality. And you're saying in humans, when we think about depression, other anxiety type disorders, it's sort of so there is a set of neurons firing in some ways, and if you can identify what might be happening, there might be some way to intervene. Is that possible?
Yes, yeah, that's exactly that we're sort of working on these days in the lab. Yeah, so we have found that this one type of neurons that we're very actively studying these days is called the corticotrophin releasing factor neurons, right? CRF neurons. So these neurons actually, exactly as you just described, is they're more active when the fish is on the dark side. So like these neurons gets activated, right? And as you know, probably these neurons, CRF neurons, is the central commander in our brain that will activate the HPA axis, right? Which is the hypothalamic pituitary adrenal axis. That's the stress axis in humans.
Yeah.
So it's quite remarkable to see that this regulation of by CRF neurons, right, is so conserved across evolution, from zebrafish to humans. And the CRF neurons have been really implicated in human anxiety as well as depressive disorders.
Yeah.
So this abnormal activation of this stress axis.
Yeah, the more I hear about these things, so the more I'm puzzled how the brain even works. I mean, it seems so finely balanced on the knife edge that it doesn't really take much to go in a direction that that has difficulties, right? So, and so you're saying anxiety as sort of a foundational issue from a neuroscience perspective might lead to a variety of. You mentioned depression, addiction, schizophrenia here. They all. You are saying fundamentally they are sort of related in some ways. Not necessarily.
Well, not necessarily. Like all these different psychiatric disorders, not necessarily they are all like closely related, right? But they're definitely share something in common. So that is they can be affected by both genetic and environmental factors, right? So like genes play a role, but also the environment plays a very important role as well in shaping these disorders. So how are they related? I think it is still yet to be understood, right? So I think human genetic studies these days have discovered quite a lot of genetic variants that are associated with these different disorders, right? And some variants actually target the same genes across different disorders. For example, schizophrenia and autism, for example, they share common genetic factors. Yeah, so they could be related in some ways.
But the activity that we see in the brain is so widespread. Are we really able to use brain imaging as a diagnostic method for any sort of disease identification?
Yeah, so I think it will be quite challenging, right? So exactly as you said, the neural activities are widespread, right? And also circuitries are very distributed in our brain, right? So we don't find like this regions is specific for this type of disease, right? So all of our behaviors are really controlled by very distributed circuits. So brain imaging will help, right? Potentially understand certain aspects of the disease. But however, the human brain imaging right now, is very is not possible to achieve the cellular resolution, right?
Yeah.
That will allow us to really understand what's going on in the brain. So zebrafish, on the other hand, allows us to do whole brain imaging at cellular resolution. Yeah, so that's something that's really very exciting. to look at to be able to link brain-wide activity and connectivity changes to certain, for example, genetic alteration in zebrafish.
Right. So the kind of the spatial detail is really difficult. Do we see any differences in the time dimension? How things change? Again, I'm thinking humans, and I'm thinking neuropsychiatric diseases. Is there any sort of temporal changes that might be diagnostic?
Mm-hmm. Are you talking about in the same individual over time?
In the images. So, going back to zebrafish, since you're doing the whole brain imaging there, are we learning anything, in terms of how the neurons are firing and how they're synchronized, how they change over time? Any of those gives us any indications?
Yes, yeah. So with respect to zebrafish imaging, yeah, we can definitely look at the temporal dynamics of the brain activity at cellular resolution. So for example, one can image the zebrafish brain activity, right? And then one can add a particular drug to the zebrafish, right? To the medium, and then you can observe how the activity might change. For example, in response to an anxiolytic compound, right, or anxiogenic compound, you can see how the brain activity now changes, right?
Yeah.
And then one can that can help us understand how these drugs potentially, alter brain-wide activity and connectivity.
So you can potentially make them bolder, right, through drugs. The zebrafish fearing the dark side of the chamber, given some agent, could be less fearful.
Yes, yeah, that's really interesting thought, Gil. Yes, that's definitely a possibility. So there are certain drugs that could affect, for example, serotonin system, right? It could definitely make them become more bold and more aggressive potentially. Yeah. So it's interesting also to mention that this dark avoidance behavior, right, is actually quite variable across the population. So I think that's something that we potentially will talk about a little bit later, is that we don't see everyone behaves exactly the same.
Right.
So some individuals seem to be a lot bolder than others. Yeah. So we see the spectrum of behavior in the population that we're really fascinated with right now, is to understand, how does evolution shape this behavior differences across individuals.
Yeah. So let's talk about that. So you have another paper, identification of brain center whose activity discriminates the choice behavior in zebrafish. Is that the one you're talking about, where you're seeing a sort of a variation?
Yeah, that paper also, we actually make use of these sort of behavior differences to try to identify a brain region that's responsible for the choice behavior. Although I want to point out that the paper that you just mentioned is actually relates to adult behavior. Right. So very interestingly, in zebrafish, their light dark preference behavior actually flips when they become adults. So when they're young, right, like when they're larva stage, they avoid dark side. But when they are older, as adults, they avoid the light side. So there's a complete switch in terms of their light dark preference.
Wow. Yeah. So zebrafish, is easier to study. Do we see this sort of humans appear to have something similar. Do we see this pattern in other biological systems?
Yes, absolutely. This light dark preference behavior actually is conserved across species, from flies, all the way to mammals.
Yeah.
So I think this behavior, the reason that it's so conserved is because, we are all earthlings, right? So we all live on this planet where it has distinct light and dark cycles, right? That the sort of the circadian rhythms allow us to, be active in the light and then, sort of go to sleep at night, right? And there's also just basically light dark have a lot of meanings to all creatures on Earth. Yeah, so it's very conserved. In fact, even in Drosophila, right, in the fruit fly Drosophila, they also have very distinct light dark preference behavior. That is also flips, when they're young versus when they're adults.
We'll take a quick break. So when we come back, we'll talk more about this, as well as sort of the hereditary aspects that we can probably learn something from as well.
Great.
Thank you. This is a Scientific Sense podcast providing unscripted conversations with leading academics and researchers on a variety of topics. If you'd like to sponsor this podcast, please reach out to info@scientificsense.com. Hi, so we are back. We were talking about zebrafish, a model that you use to look at the brain, because obviously they provide some interesting ways to get the data, and it is a lot simpler than human brains. But there's certain behaviors that appear to be similar. In the case of the zebrafish, the young zebrafish seems to have a fear of dark places, just like humans. And as they grow up, they appear to like it and have an aversion to the lighter side. One of the hypotheses you have, if I understand it, so is that the anxiety is sort of the reason why this might happen. You have another paper in 2016. You're looking at sort of the identification of environmental stressors and validation of light preferences as a measure of anxiety in larval zebrafish. And you look at variety of stressors, stress stimuli such as heat, cold, ultraviolet, mechanical disturbance, and social isolation. So what do you find here in this study?
Yes. So in that study, we're curious to know, whether the behavior, this light dark preference behavior in larval zebrafish, right, are indeed sort of fear and anxiety related, right? So previously, we and others have shown that anxiolytic drugs, right, can modify this behavior, which sort of provide some pharmacological evidence that this behavior may be anxiety related, right? So in this particular study, we kind of wanted to know, sort of on the flip side, can we make this behavior worse, right? By, treat the larval zebrafish with anxiogenic stimuli, right? Or stressors in this case. So if it does, modify the behavior, then we can, be more confident that this behavior is somehow anxiety related. Yeah, so that's sort of what this paper, this simple paper about.
Okay, and so what you find is that these stressors actually make the behavior more pronounced. Is that what you find?
Yes, exactly. Yeah, so that's exactly what we found. So we found that when we heat zebrafish up, right, basically putting them in a hotter water than they usually like, and then we introduce them into this light dark chamber, and they will show much more pronounced dark avoidance behavior. And the same goes with cold. When we put this larval zebrafish in a colder water, right, than what they usually desire, and then test the behavior, it will also the avoidance behavior becomes more pronounced, and among some other stimuli that we also have tested as well. So this experiment, one thing we want to determine is whether the behavior is anxiety related. Another thing is we want to find other stimuli that can enhance this behavior, and then try to understand how this stimuli actually modify the circuit that leads to enhanced behavior.
Yeah. It's always difficult to extrapolate from zebrafish to human, but does it have some implications for humans as well? So in the case of the human, I think environmental stressors, there is a whole plethora of environmental stressors, from chemicals to a variety of things, right? So do you think those stressors could have an impact on the neuropsychiatric diseases that humans have?
Yeah. So I think definitely, a lot of these human and psychiatric diseases there have a significant environmental contributions. Chemicals being one possibility, and also early life stress being another possibility, right? So a lot of these work with genes, that could precipitate phenotypes or symptoms in the vulnerable human beings.
Yeah, it obviously has implications for healthcare more broadly. So, our general tendency is to treat after a disease shows up, but it is much less costly and much more effective if you can intervene in a preventative fashion, right? And perhaps we are beginning to do that on the physical side, but doesn't look like we have done a lot on the mental side, or mental disease side, in terms of proactive interventions early on.
Yeah, I agree with you. I think I guess it's challenging in a way, right? So in order to prevent or intervene, we have to be able to recognize some telltale signs early on, right? So that we can identify the sort of the sensitive or the vulnerable populations, right?
Yeah.
But I don't, I'm not a doctor, human doctor, so per se. So I'm not sure how that area has been developed in terms of early diagnosis, right?
Yeah.
Of psychiatric vulnerabilities. Yeah.
Yeah, I mean, obviously, it's very difficult to extrapolate. But what you're finding here is that there is a behavior that is potentially driven by anxiety, and if there are environmental stressors present that increases anxiety, that behavior is amplified. And then obviously in a simple system like Zebrafish, it seems to make intuitive sense, right? So so could you extend these types of things to, let's say, a mouse model or something like that, to a bigger system?
Yeah, I think it's definitely I think these type of studies have been carried out in like a mammalian models such as mice and rats, right? So, but each system have its own strengths and the sort of limitations as well. So the zebrafish system, the limitation being it's even though it's a vertebrate, right? It's not a mammal. So in a way that it's less close to humans, right? Yeah. But zebrafish have a lot of strengths that rodent systems don't have. For example, the brain, the rodent brain is very complex, much more complex than the larva zebrafish brain that we are studying, right? So the simple systems here offers actually a tremendous advantage to be able to really understand the behavior and the circuitry at a cellular and molecular levels. So I think that's the reason. I think what we use zebrafish, and that's what we think zebrafish can contribute to the to this question. And also the zebrafish is very prolific, so we can get lots of these little fishes for experiments. Yeah, so we could do a lot of high throughput behavior phenotyping and then genotyping experiments.
Yeah, yeah, and the objective function in the brain is somewhat similar too, I would imagine. Food, predator, and there aren't too many factors that so the noise you have when you image the brain is potentially lower too, right? In the zebrafish, potentially, I don't know.
Mm-hmm. Yeah, I think the in general the circuitry is probably simpler. I would say, for example, let's just take this particular nuclei in the vertebrate brain as example, right? So there's a nuclei called locus coeruleus. So it's a noradrenergic center in the brain, right? So it's in all vertebrate brains. So in zebrafish, so this locus coeruleus noradrenergic center only have 3 to 10 neurons, whereas in mammals, it's probably at least 1,000-fold more, if not even more, right? So it just, the complexity is definitely daunting. Yeah. So in the mammalian brains.
Yeah, so more broadly, Sue, I know that you do a lot of work in this area. More broadly, are we finding that you can replicate complex behaviors and complex objective functions with a very few number of neurons? Is that is that statement true? I mean, a few 100,000 neurons, entire brain of a zebrafish, and you're saying certain aspects of that brain is actually few neurons that is making the decisions. It seems to me that is sort of a different feel when we think about the brain as so complex, right? Perhaps the brain is not that complex, is it?
Yeah, that's a very interesting thought. So I think, even when we look at 100,000 neurons, we are already faced with a quite daunting challenge to try to understand how these 100,000 neurons are talking to one another. Because when we perform neuroactivity imaging, for example, we just see, cells here, like fireworks, like goes off here and goes off there, right? And then we have tried to make sense of what this this firing activity is all about. So I think I think brain scaled up, right, as we as species have evolved on Earth, right? So there must be a lot of added on functions, right? So for example, human brain can do so much more, right, than a zebrafish brain can do, right? But a zebrafish brain will allow us to really look at some tractable behaviors. In this case, we're interested in this choice behavior. How a simpler brain actually make decisions, make choices, right? So once we can understand a simpler brain in a greater detail, right, at cellular molecular levels, then it will provide potentially a framework, or maybe potentially some type of theoretical framework or principles that will help us understand this enormously complex brain like our own.
Yeah, yeah. The brain appears to be highly optimized. It is dealing with some very, very harsh constraints in terms of space, in terms of power. And so how the brain works is quite different from where the AI research is heading. AI research is going in the direction of let's throw more silicon at the problem, and if you will get a better answer. The brain is almost exactly the opposite. It is really about optimization and resource constraint. rather than throwing more resources at the problem, right?
Yeah, I think so. I agree. Yeah, I think the brain is extremely efficient. So it's called, I think this, a complex network, right? Small world network, the human brain. Or as a matter of fact, all brains actually follow this small world network rules. Yeah.
Yeah. You have another paper, 2017, Heritable natural variation of an anxiety-like behavior in larval zebrafish. And so you went into this looking at if you can actually see um, hereditary aspects to this anxiety-like behavior in zebrafish. Did you find it?
Yeah, so yeah, let me give you a little bit introduction about what this work is about, right? So far we have discussed this light dark preference behavior, right, in larva zebrafish, and in general we see larva zebrafish avoid dark, right? So they find dark side to be aversive, right? However, so that's on average, right? So usually we do a behavior analysis on like 20 to 50 individuals, right? And then we measure, we develop a preference index, right? So basically how much time they spend in the light side versus dark side, right? And then we just develop an index, subtract the percent time in the light side minus the percent time in the dark side, right? And then we average them, right? So then we basically get this population average, right? Which suggests that larva zebrafish find dark side aversive, right? Have dark avoidance behavior. However, when we look at on the individual level, right, we see quite amazing phenomenon, if you will. So you can see some individuals, right? They are extremely dark aversive. So meaning that, during the entire behavior recording period, they never even, cross the boundary ever once. They spend all their time avoiding dark side, right? And then we also see on the other end of the spectrum, individuals who doesn't seem to even care about, light or dark side. They just freely explore both sides as if they have no fear, as if they're just, completely very bold, right? So we see this distribution across the population. So this reminds me of, behavior spectrum, right? Even in humans, we have some very brave, human beings, right? We also have some very, fearful, scared human beings, right? So the same is happening in fish, seems like, so that really, interested me. I wanted to understand what determines these differences, right? So one question obviously is it genetic, right? Or is it purely non-genetic, right? So in order to address that question, the experiment that we did is to basically selectively breed individuals, right, who are extremely exploratory, right? And then selectively breed the individuals who are extremely risk aversive, right? Never go to the dark side, right? And then we look at their babies, their next generation.
Yeah.
So basically we ask, is this very bold individuals, do they also give next generation also very bold, right? And then the ones that are really risk aversive, dark aversive, do they give individuals that are also risk aversive and dark aversive, right? So and then our answer is yes. That we did this. We discovered there is genetic components to this behavior differences across the population. Yeah.
Yeah, and so you describe in this paper you have sort of three buckets: medium dark aversion, strong dark aversion, and variable.
Yes. Go ahead.
And so you basically go back and breed the members in those buckets, and then look at the next generation if they're showing sort of similar characteristics, right?
Exactly. Yes.
And
Yes. Yeah.
I think there's a strong correlation. So you would conclude that there is a strong genetic component to darker version?
Yes, I don't know how strong it is, but it's completely detectable, right? We can detect heritability of these behavior variations. Yeah. So suggesting there are genetic contributions.
And the neuropsychiatric diseases, obviously there are a whole spectrum of those diseases. My understanding is that there is also a fairly strong genetic component there, right, in humans?
Yes, I think so. So it varies from disease to disease, right? But definitely there are very detectable genetic contributions, yeah, to like schizophrenia, to anxiety disorders, and so on. Yeah.
Yeah. And it's probably too early, and more studies and maybe different models. But would you say so? Is anxiety sort of the parameter that you can look at if you titrate up and titrate down anxiety? You can then predict what the behavior, the changes in behaviors might be. Are we there for zebrafish? In other words, you did this in one study where you have this environmental stressors, and you showed that the higher the environmental stressors, the more amplified the behavior is, right?
Yes.
And so, but we don't know. We cannot say if environmental stressors, the ones that you use like heat and cold, uh, lead to anxiety. But are there more direct ways you can increase the anxiety of the zebrafish? And try to answer the question: is it anxiety that is leading to this behavior?
I see. Okay, so yeah, so anxiety is basically quite loaded term, right?
Yeah,
So how do we directly increase anxiety? I mean, it's not. I mean, anxiety is actually read out as a behavior here,
yeah,
right? Because in animals, in all animals, right, we cannot directly ask them, right, how anxious do you feel, right? Because in humans we can, right, we can just talk to the person and then say, how do you feel, right? And then the person will be able to tell us, right?
right.
But we cannot communicate with animals, so the only way we can read out sort of their anxiety level, quote, right, is through behavior monitoring, right? So the way that we can, how do we change their directly change their anxiety, is very difficult to know, right? So the way that we have been doing is basically using, for example, drugs, right, that we known to alter anxiety in humans,
Yeah,
right, or stressors that we kind of know that will likely to be anxiogenic, right?
right.
So we kind of just sort of still using this type of approaches. Yeah.
Yeah, yeah. It's a complex question. So since you're finding a genetic component, I was wondering if there is again some correlations to the visual systems and other aspects that lead to this behavior. But on the other hand, since you're demonstrating that environmental stressors amplify the behavior, that is sort of indicative of the anxiety aspect, right? I mean, that is probably the best you could do to answer that question.
Yeah, so I think you bring out a really, really important point is, what, for example, what is really underlining, right, these differences in individuals' behavior, right? Because as I just, told you that across the population, we can see some individuals that are seem to be extremely, indifferent, right, to the light and dark side. And there are some individuals who are extremely dark aversive, right?
Yeah.
So what's the genetics that underlie these behavior differences is something that we're extremely interested in finding out, right? So as you mentioned, one possibility could simply be, somehow their visual system is different, right? So allows them to somehow, see the behavior chamber differently, right?
Right.
Whereas everything else inside their brain is the same, identical, right? I mean, it is hard to imagine that this would be the case however, we cannot exclude that possibility at this point.
Yeah.
Right. So because when we look at these individuals, right, they don't look different. In terms of just looking at their morphology, right? Their eyes look perfect.
Yeah.
They seem like to be able to find food perfectly. So they don't have problems, finding food, right? Which suggests that their visions are not that different, right? I mean, it's not like they're blinded, so they couldn't, they become indifferent, right? So it's not the case. Yeah, it's not the case that they're blind. So what we're extremely interested in finding the genetic underpinnings of this behavior difference, because we think this could be a very interesting entry point to really understand this type of behaviors in other species as well. Because if you think about, right, even as human beings, right, we are all have different level of risk aversion as well, right? So certain individuals are extremely exploratory, right?
Yeah.
So like Elon Musk, for example, wanted to go to Mars, right? Extremely exploratory. And there's also individuals who are very risk aversive, right? So what determines this type of traits, right? Could we learn something from studying zebrafish, right? Is something that we're very interested in finding out.
Yeah. So the other thing I was wondering about, is, do you find if you take the ratio, so you have three buckets: medium dark aversion, strong dark aversion, and variable dark aversion, MDA,
Mm-hmm.
SDA, and VDA
Mm-hmm.
buckets. If you take the ratio of members in those buckets, in various, sort of sampling experiments you have done, do you see that ratio to be generally the same, or it's highly variant? In other words, if you take a random sample of 100, you almost always see 30 in MDA and 40 in SDA. I'm just making this up, and 20 in VDA. Do you see any sort of stability there in ratios?
Yeah, that's a really good question. So in fact, we see a majority, right, of the population are in the MDA bucket.
Okay.
So they have like a medial level of dark aversion, whereas the extremely dark aversive or the extremely exploratory fishes are on the smaller percentage side. So it I think it makes sense. So I've been thinking about why the fish wanted to have this behavior variation, right?
Yeah.
So I think it's probably programmed evolutionarily.
Yeah.
Right. If you think about it, these fish living in the wild, right? They could face many different potential environmental conditions, right?
Yeah.
So in certain places, perhaps there'll be a lot of predators, right? In which case, the VDA buckets will probably be gone, right? Be selected against, right?
Yeah.
Whereas the SDA hopefully will carry the population, right? Forward. Whereas in another type of situation, for example, food scarcity, right? Like in some type of famine conditions, right? You would think that all the SDA would probably be eliminated, right? Because they're too scared to go out to look for food, right? Whereas the VDA bucket potentially will carry the population forward, right?
Yeah.
So I think having this behavior variation in the population really will allow them to become more resilient to the environmental changes that allow the species to be carried forward.
Yeah, that's very interesting. So the MDA bucket, the both ends of that spectrum. So you have sort of a distribution. Both ends of that spectrum has higher risk, and in different contexts, they're more likely to get eliminated. So MDA has the highest membership. In some sense, you could think about this sort of societal evolution, right? I was thinking about humans. The really adventurous types of humans, that ratio between very adventurous to average, that ratio cannot be too high. Otherwise, we would have been, Homo sapiens would have been all eaten up by lions and tigers very early on.
Right.
But you need to have some percentage of those people. Otherwise, you would have been stuck in Africa forever, right? So So there's sort of an optimum ratio, it seems to me, between those buckets that make the society to evolve optimally. Do you see that way?
Yeah, I do. I think you're absolutely right. I would imagine so, right? Even with Homo sapiens. Yeah. If you think about it, the adventurous type, right, will be very, very important, right? In the face of some type of conflicts, right? Whereas the sort of, yeah, as you mentioned, if we don't have these adventurous type, we probably will not be able to explore the entire Earth, right?
Yeah, in other words, we need a few Elon Musks, but we don't need too many of them.
That's right. Yeah, you're right. If we have too many of them, potentially we would also it will also become a problem. Yeah.
Yeah, and I don't know, cross sectionally, I think that it will be very interesting to see if that ratio is similar across different animals, different animals, right? And clearly the threats that you face are very different for different animals, and so presumably it's different for different species. But my gut tells me that there might be some sort of an optimum ratio there that we may be able to find.
Yeah, so I think oftentimes, if we look at any type of behavior, right?
Yeah,
So we would oftentimes the extreme end, right? Usually are represented by smaller individuals, smaller percentage of individuals, right? Because the sort of the distribution. If you look at a distribution of a behavior, right?
right.
So majority would be kind of in the middle, right?
Yeah,
And then the extreme ends will be represented by fewer
right.
individuals, but that could change depending on the environment, right? So if in the like really if we shift the environmental conditions, right, that could potentially favor one end, right, versus the other end. Then the population ratio could evolve very differently.
Yeah, exactly. This is not in the paper, but so as you say, as the environment changes, that ratio has to change. And if it doesn't change, you can get extinct, possibly, right?
Yes.
And so, yeah, that's sort of an interesting question from an evolutionary perspective. Um, the distribution that we see today. I mean, they're talking going back to thinking about humans. The distribution that we see today is a function of history, and that may not be ideal, right, going forward. For instance, all the domesticated animals, appear to show lower brain size. They're getting dumber and dumber over time because they don't really have much challenges. They're basically depending on humans to get everything done. So they don't really need the distribution they used to have when they were in the wild, right? It's a different thing. Similarly, humans are changing as well. So I wonder how things would be in the future for humans.
That's a very interesting question. So yeah, I remember there was a movie about that.
Yeah, I sometimes think it could be domesticated by machines in case humans don't have much use for their brains ultimately.
Yeah. So that's hopefully that doesn't happen, right? But that could be a potential danger.
Yeah, yeah,
So I guess since we believe that we want to think about, right,
right.
things and doing a lot of intellectual work, right? That hopefully will keep our brain active, and not sort of retract.
Yeah. Well, it's just speculation, but one could envision situations you can even delegate research to machines in the future, uh, which would then make humans pretty useless. Uh, So in conclusion, I know that you're doing a lot of work in this area. Zebrafish appears to have a lot of very interesting advantages and characteristics to study this. So as you look forward, 5 years, where else do you think you will take the Zebrafish model in terms of understanding? This is really about behaviors, diseases, those types of things from a brain perspective. Where else do you think you could take it?
Mm-hmm. Yeah, so that's a really good question. So in terms of this behavior that we're studying, this light-dark preference behavior, I'm extremely interested in knowing what are the genetic landscape, right, that underlines these individual differences in the behavior. So we're taking a variety of molecular, cellular, and genetic approaches to try to find the genes and also the single nucleotide polymorphisms that are present in this population to see, to try to understand which type of genetic changes might underline these behavior variations that we have observed. And also, ultimately, we also want to know if these type of genetic variations that we detect in zebrafish will also be, present in other species, including humans, possibly, right? So then maybe one day we can really understand, the differences between explorers versus, right, like caretakers, for example, right? So both of those are very, very, important tasks, but we don't understand how they're genetically determined, right? So maybe one day we will be able to understand that. That's something that I would be really thrilled to be able to achieve maybe in 5 years.
Yeah. So sort of the genetic and almost like the molecular level, understanding what causes it, and if you can understand it, then perhaps you can. I'm thinking about diseases now. If you can show a connection between that to certain diseases, then perhaps there is intervention there, not just chemically but genetically.
Yes, that would be one possible connection to diseases. So, for example, the individuals that we were just talking about, the SDA bucket, right? So those individuals, we postulate that they could be more sensitive to environmental stress, right? So if we do, we could test this hypothesis by apply stressors to SDA bucket versus a VDA bucket, and then see if some type of pathological state can arise, right? Because anxiety, normal dose of anxiety is essential, right, for self-protection, but it's the pathological state of anxiety that's unwanted, right?
Yeah.
So we could potentially use these different buckets of individuals to test the hypothesis.
Yeah, the beauty of the zebrafish model is that, as you mentioned, there are plenty available, and the brains can be really looked at a very detailed level. So, yeah, it's exciting research. So, thanks so much for time with me.
Thank you so much.
Thank you.
This is a Scientific Sense podcast providing unscripted conversations with leading academics and researchers on a variety of topics. If you'd like to sponsor this podcast, please reach out to info@scientificsense.com.